Electron carriers are compounds that shuttle around high-energy electrons via redox reactions, the coordinating states of oxidation and reduction respectively, losing and gaining these negatively charged particles. For instance, one principal electron carrier is nicotinamide adenine dinucleotide, or NAD+, named so because at the first carbon position, one ribose ring has an adenine base, while the other has a nicotinamide. At the fifth carbon position, two phosphate groups join these two nucleotides. During metabolism, NAD+, as a coenzyme, binds to an enzyme such as dehydrogenase and acts as an oxidizing agent, removing two hydrogen protons along with a pair of electrons from the reactant. The electrons are then transferred to the positively charged nitrogen, and the hydrogen attaches to the opposite carbon, reducing NAD+ into NADH. In the end, the extra hydrogen proton is left in the cytoplasm, and NADH shuttles its electrons to the mitochondrial membranes, where they enter the electron transport chain, and the carriers can continue to undergo redox reactions. The complete oxidation of one glucose molecule yields 30 to 32 ATP molecules. It starts with glycolysis yielding 2 ATPs and 2 NADHs, followed by the oxidation of pyruvate producing 2 NADHs. Next, the citric acid cycle yields 6 NADHs, 2 FADH2s, and 2 ATPs. The NADH and FADH2 molecules are oxidized in the electron transport chain to produce 26 or 28 ATPs. This range of ATP is approximate for three reasons. First, the electron carriers indirectly produce ATP by pumping hydrogen ions into the intermembrane space, producing theoretical fractions of 2.5 and 1.5 ATP molecules. Second, NADH produced in glycolysis cannot pass through the mitochondrial membrane, so high-energy electrons are transferred to other electron carriers within the mitochondria, and depending on the cell types, produce FADH2 or NADH, yielding 1.5 or 2.5 ATPs. Third, the energy produced by respiration is also used to power other activities, like the transport of pyruvate through the mitochondrial membrane. Glucose, though a simple energy source, is not common in the diets of humans and other animals, so they obtain most of their energy from the digestion of carbohydrates like disaccharides and polysaccharides, proteins, and lipids. A polysaccharide like glycogen, a chain of several glucose molecules, is first hydrolyzed into glucose and fed into glycolysis to produce pyruvate and high-energy molecules. A disaccharide like sucrose, a sugar found in most fruits and vegetables, is broken down into two simpler sugars, fructose and glucose, and both are then catabolized in the glycolytic pathway. Proteins are first broken down into constituent amino acids. While most amino acids are recycled to build new proteins, others can undergo deamination and enter the glycolysis and citric acid cycle at different stages. Lipids, like triglycerides, are catabolized into glycerol and fatty acids. The glycerol component is converted to a phosphorylated form that can enter glycolysis, while the fatty acid chains are converted by beta oxidation to acetyl-CoA, which then enters the citric acid cycle.